So when we last left off in the last video, we were talking about meiosis and how meiosis is a two-part process, and that is all true. But I want to talk to you about how males and females, they go through this process slightly differently. Um, Males, they go through the process kind of basically exactly as I've described it to you. And they start out with one parent cell, they end up with 4 daughter cells. Those 4 daughter cells are now called sperm. That's how males do it. But females, they have to make eggs, and eggs are, relatively speaking, large cells. And especially if you compare it to the size of a sperm, like this is an egg, and that's the sperm right there. So males produce lots and lots of small things, and females can only produce one large thing. So how do they go through this process? Starting out with this parent cell right here, and this parent cell is diploid. How do you know it's diploid? Because it's got its homologs. So here's the blue homologs and the green homologs. And in the end, we're going to end up with one cell that's haploid. So how do we get there? We're going to go through replication, right? So interphase. And here we are after replication and after telophase 1. And notice that the cytokinesis happens unevenly. And what happens with cytokinesis is that only the nucleus gets put into this egg, this cell over here. And all the rest of the cell contents, all the rest of the cytoplasm, the mitochondria and the smooth ER and the rough ER and the Golgi and all of those other things, that gets put here. So this is a functioning cell with all of the stuff from the cytoplasm in there. This is just a nucleus, and a nucleus, it's useless without the cell's machinery, and so that dies. This structure here is called a polar body. So a polar body refers to the small nucleus that's gotten rid of half of the genetic information. That's the polar body, and again, that dies. So only this cell right here continues with meiosis, and we can follow it up to here. There it is in meiosis, and we're going to go through meiosis 2. So we're going to go, and with meiosis 1 and going into meiosis 2, this is telophase 2 right here, and I think this might be a typo. I think that's supposed to say meiosis 2. And we were going through the cell division process again, but we also end up with another very small nucleus and a rather large cell with all the stuff in the cytoplasm. And this is what becomes the egg. It became haploid here, but it lost its sister chromatid here, so this is the final product, the haploid final product. And this polar body, it dies. So it degrades. And that's how you end up with one large egg. So males and females do make their gametes differently. There's a couple other things that I want to talk to you about with meiosis, including, like, I want to focus in on that crossing over thing. So crossing over is a really big deal. It doesn't seem like it at first. It's just like, well, why is this maternal chromosome swapping, DNA with this paternal chromosome? And we'll get to the whys in a second. But to start out with the hows, remember this is the maternal and this is the paternal. They're homologous. Homologous chromosomes are also called homologs, and those are the sister chromatids. So notice that in the sister chromatids, both of them, this isn't swapping with that one. It's just like one of the sister chromatids from each homolog cross over and trade bits. And so you end up having this chromosome right here not having sister chromatids that look exactly the same. This one looks like the original maternal chromosome, but this one looks like mostly maternal with a bit of paternal. Same thing over here. This is all the original paternal, and this one here is mostly paternal with a little bit of maternal. And so what you end up with, and this is the really interesting and cool thing, is when these all separate out into their 4 final cells, no single chromosome is exactly the same. So when we were looking back here at why the brothers of the Kennedy family didn't look exactly the same as each other, a big part of that is crossing over. So that how this bit crosses over, how you get the maternal, did you get this maternal chromosome or did you get this one? Did you get this paternal chromosome or this one? That adds variation into the offspring. And variation is one of the big deals about meiosis. It's actually one of the things that meiosis functions as, is to increase variation in the next generation. So, so we could think about the costs and benefits of sexual reproduction, and we're going to do that. So there's a few costs and benefits, but let's start out, before we get into the costs and benefits here, let's talk about other sources of variation. Crossing over is the one that I just spent a lot of time on, right? So these are all sibling kittens and they don't all look alike. Why do they not look alike? Crossing over is a big deal, right? Maybe one of the kittens got that bit, but the other kitten got that bit, right? So that's one reason. Another reason is because, of course, your parents aren't the same. And because your parents aren't the same, they're giving you different versions. Maybe your mom has red hair and your dad has brown hair, or maybe your mom has straight hair and your dad has curly hair, or something along those lines. They are different, and so that's another source of variation. And then there's this third thing that's really important, but also very hard to wrap your mind around. And your book calls it reassortment of homologs. That's what it calls it in this chapter, reassortment of homologs. In the next chapter, it calls the same exact thing independent assortment. So I'm going to use those terms interchangeably, reassortment of homologs and independent assortment. They're basically the same thing. And I want to take some time to explain this to you, but it's really hard with this static picture. So I'm going to once again encourage you to watch the other videos that have more animation associated with them to understand how important this reassortment is, also known as independent assortment. So here's the thing, and again, we'll be dealing with this in lab, and that is that, if you recall, when we were looking back up here for, oops, sorry about that, for how these chromosomes line up on the metaphase plate or the equator during metaphase, you notice that this light green one is kind of near the North Pole, the dark blue one is near the North Pole, and this lighter yellow one is near the North Pole. So that's not set in stone. Like, this is where the reassortment concept comes from, is that we can randomly assign which one is headed towards the North Pole. Maybe in the next cell that this happens in, the dark green one will be up here with the dark blue one and the light yellow one. Or maybe if we do this a little bit differently, it could be the dark green one with the light yellow one, but this would be the light blue one. There's all different combinations of the way these can line up on the metaphase plate, or also known as the equator. So again, we call that reassortment of homologs. Again, it's a weird term. I think independent assortment is a little bit of a better term, but this is what we're going with because your book brings it up. So, and again, we will deal with this in lab, and we'll even do a demo in lecture. So, stand by on that. So the question on this slide was, what are the costs and benefits of sexual reproduction? And so I haven't answered that yet. Let's take a look here to see if we can come up with an answer. One thing that we do know are things like bacteria, they reproduce asexually. We know some plants can reproduce asexually, but most plants reproduce sexually. Animals, for the vast majority of them, they're reproducing sexually. Is one method of reproduction better than another? One thing we do know is that the environment that everything lives in is gradually changing, and it changes from one generation to the next. Sometimes this change can happen really fast. Sometimes it happens much more slowly. If you're an organism that has a set of genes that work really well in the environment that you're in, then that's great. You're going to survive that environment. But what happens if the environment changes? Well, if you're reproducing asexually, then you will have the same exact genes as your parent. And if the environment changes, then you might not have the genes you need to survive that environment. If the environment is changing and sexual reproduction is happening, on the other hand, then there's more variation in those offspring, and maybe some of them are going to be able to match the environment a little bit better. It's kind of like buying a lottery ticket. You know, the more tickets you buy, the more likely that one of them you have as a winner, and that's kind of like sexual reproduction. On the other hand, there's a lot of costs involved in sexual reproduction. I mean, let's face it, finding a mate can be dangerous. In the case of the black widow spider, it can be quite deadly. So that's not good. Although it's not good for the male, I guess it's great for the female because she now has a nutritious meal to feed her young with. Other things that can be bad is that when you're mating, you're basically not on the lookout for predators. So maybe you could be tracked down by a predator. There's also diseases associated with sex, as we're familiar with. There's other things that can go wrong during mating and reproduction. There's lots of risks. In addition, besides all these risks, if this sexually reproducing organism right here, the spider, if she's like really well adapted to her environment and her environment isn't changing, then she's only getting to pass on half of her genes. And that's, that's a cost. Especially if maybe this one wasn't doing quite so well in its environment, it still gets to pass on half of its genes. Whereas with asexual reproduction, if the environment's not changing, then hey, they can reproduce really, really fast. They don't have any of those costs of sexual reproduction. There's no venereal disease associated with bacteria. Okay, they cause it, but they don't get it from sex because they don't have sex. So those are some definite costs and benefits. Fast reproduction in asexual and all the costs there. So which one's better? Sexual or asexual? Honestly, it depends on the environment, and environments change. And so that's one of the reasons that sexual reproduction is so widespread, because environments do change. So a lot of take-home message here from this. There's two fundamentally different ways that eukaryotic organisms reproduce, and that's mitosis and meiosis, which correspond to sexual and asexual. Mitosis is the asexual form of it, and meiosis is the sexual. Sexual reproduction can be fast and efficient. This should say, this is a typo, I'm very, very sorry. This should say asexual. Asexual reproduction can be fast and efficient, but offspring are identical. And whatever happens in that environment change, it could be a disadvantage to the organism. Sexual production is slower, costs a lot more energy, takes a lot more risk, but your offspring are genetically different from each other and from the parent, and that can be good for the long term. We're going to kind of wrap up our lecture really focusing on two more things. One is, how is sex determined? And the second is, what can go wrong with cell division? So starting out here with how is sex determined, and we're going to start out talking about humans. Humans determine sex based on a specific pair of chromosomes called the sex chromosomes. And those chromosomes have names. The names for the chromosomes are X and Y. And you can kind of see in this picture why they're called X and Y. It's a little misleading, though. I want you to understand most chromosomes look like this X chromosome. It's the Y chromosome that's a little strange. It has lost large chunks of the rest of the chromosome, and so it kind of looks a little bit more like a Y, like the letter Y, which is why we name them X and Y. But again, most chromosomes do look like this X chromosome, so it's a little misleading. This is a male here. Males have one X chromosome and one Y chromosome. Females would have two X chromosomes, one from mom and one from dad, right? So if dad's going to have a baby girl, he's going to give this one. If dad's going to have a baby boy, he's going to give this one. So that's how sex is determined in humans and most mammals and lots of different species. But that's not the only way, and we'll get some exceptions to that in a couple of slides. For now, let's just again focus on how sex is determined in humans and realize that the female in this process does not determine the sex of the offspring. It's the male that determines the sex of the offspring. Females have two X chromosomes. It doesn't matter whether she's having a boy or a girl. She's going to give one of those X chromosomes to her offspring. So if the egg is fertilized by this X chromosome hanging out in the sperm, you'll have a girl, and if you got a Y chromosome hanging out in the cell, you'll have a boy. So the male determines the offspring, the sex of the offspring. One of the things that we do know about the Y chromosome is that it is necessary for there's no information on the Y chromosome that is absolutely necessary to grow a human being. So it's possible to have a human being have only one X chromosome and live a normal, healthy life. It's not necessary, but it is necessary. The Y chromosome is necessary if you're going to have a male. The main information that you're going to find on the Y chromosome is something called the SRY, and the SRY basically makes sure that testosterone is made, and secondary sexual characteristics are made from that testosterone. They also have genes on them, like, for example, hairy ears. Females do not have hairy ears. Some males do have hairy ears. That's because they happen to have a version on the Y chromosome that calls for hairy ears. I mean, really not strictly necessary, but still exists. In other species, there's different ways that sex is determined. Well, one of the things that I want you to know is that not all species have different sexes. Some species exist as hermaphrodites, and that simply means that one organism can make both male and female gametes, can make both eggs and sperm. Earthworms, snails, in fact, most plants can make both eggs and sperm. So we call them hermaphrodites. Sometimes you use that word hermaphrodites to describe humans, but it's literally not true. It's not an appropriate term to use for a human, because we do define hermaphrodites as whether or not they can make sperm and eggs. And in a human, if a person has secondary sexual characteristics that look kind of like some of them are male and some of them are female, for example, it depends on what the gonads are. So do they have ovaries or do they have testes? Or are they somewhere in the middle? If like the ovaries, and you should watch this video that I put on Blackboard about how males and females are different and how they're made. But ovaries become testes in a male, so everybody starts out as female. And so if you only have this one structure, ovaries, and they stay as ovaries, then you're female. And if they descend and leave the outside of the body, then you're a male. You can't have both. So that's a hermaphrodite. When it comes to other species, like birds, it's actually the female that determines the sex of the offspring, not the male. So instead of having X and Y, we use the terms Z and W, and read a little bit more in your book about this ZW thing. It's really, really cool. But the outcome of it is, in humans, the male determines the sex of the offspring. In birds, the female determines the sex of the offspring. In birds, females have the two different types of chromosomes. They're called Z and W. And in the male, they have two of the same chromosomes. In ants and bees and wasps, whether you're male or female has to do with how many chromosomes you have. If you have information from both your mom and your dad, you will be a female. If you just, and this is so wild, if you just have information from your mom, you'll be a male. Because in ants and bees and wasps, it's not about which chromosome you have, it's how many chromosomes you have. Males are haploid and females are diploid. That's wild. Even more wild is what happens in turtles. Because turtles, it's not about chromosome number, it's not about, X and Y or Z and W, it's all about temperature. So what temperature you are kind of conceived at and delivered at, like how the egg is developing, that determines whether or not you're male or female. And we're going to talk about how that works in this next little section on how, can the environment determine the sex of a turtle's offspring? So we didn't actually know this until the 1960s when this researcher, Madeline Charnier, she saw that the sex ratio in lizards, which are related to turtles, was influenced by the environment. She saw that when it was warmer out, there were more females, and when it was cooler out, there were more males. So what she did was brought two sets of turtle eggs into the lab at different temperatures, at 25 degrees C and at 30 degrees C. At 25 degrees C, when they hatched, they were all male, and at 30 degrees C, they were all female. Now, in this experiment, the temperatures were actually held constant at 25 degrees or 30 degrees. Clearly, that's not what happens in nature. Clearly you get temperatures that fluctuate throughout the day or throughout a season. So in a next experiment that they did, they kind of took these daily fluctuations in temperature into account, and they had this daily fluctuation. So they incubated the eggs in the lab, and they also did something similar outside in the natural habitat, where one nest was in the shade and one was in the sun. And the same thing happened. The overall average temperature, if it was cooler, you'd get more males, and if it was warmer, you'd get more females. So that's really, really cool. Again, not discovered until the 1960s. So thinking about these results, would you be confident that incubation temperatures influence the sex of turtles? Like, would you be confident in that? This is just an interesting question, but here's an even more interesting question. How could climate change have an impact on turtle populations? That's something that is happening and that we need to think about. In the next 100 years, we're expecting to see average global temperatures rise by 2 or 3 degrees. And remember, it was just 5 degrees that made all males versus all females. That's a big deal. So, and it's not just turtles. Remember that we have the same kind of thing happening in lizards and so forth. So just keep that in mind. The last thing that we're going to talk about is problems that might happen during sexual reproduction. And this is an example. This first one that we're stepping through is an example of Down syndrome. And so I'm just going to show you this thing. This picture right here is called a karyotype. And with a karyotype, what you have is just a picture of chromosomes, and these chromosomes have been arranged next to each other, next to their homologue. So I already told you about how there's some chromosomes called X and Y, and this karyotype has 2 X chromosomes, so that makes this a female. But all of the other chromosomes, they have numbers associated with them. So there's chromosome number 1, number 2, goes all the way through number 22. That's your chromosome set. And they're arranged, somebody actually has to do this job of moving these chromosomes around so they look like they're arranged well. and they take this picture, and this is called the karyotype. So, so this is a karyotype of a normal human being, but as people age, and we have good data on what happens if mothers age, that you're more and more likely to make a mistake with how you separate your chromosomes from each other, and you might do that improperly. So, so you can actually test to see whether or not you have properly or improperly made your eggs, but only after you have a fetus growing inside of you. So you could test for these abnormalities that I'm about to talk to you about through amniocentesis, which like a needle is put into the belly of the pregnant mother and you take samples of the amniotic fluid, or you can do it by chorionic villus sampling, and that's where the cells are taken from the placenta instead of from the amniotic fluid. So those are the two options for figuring out whether or not you have made some sort of chromosomal abnormality or mutation. This is a chromosomal mutation in making your gametes. So what happens if you do make this mistake? And this is an example of trisomy 21, which is also known as Down syndrome, named after Dr. John Langdon Down back in 1866. And what he just basically described the syndrome. But what we know now is that by looking, and he didn't, I mean, there were no pictures of chromosomes back then, but we do know now that chromosome number 21, in the case of Down syndrome, there's actually 1, 2, 3 copies of it instead of the normal 2 copies. So you get this extra copy of chromosome number 21. And this kind of mutation happens in about 1 in every 1,000 children. You don't have to be of advanced age to have this happen to you. It's just more likely to happen to your child if you conceive at a later age. So Down syndrome. And so how does something like Down syndrome, where you get an extra chromosome, how does that happen? And it happens through this process called non-disjunction. So non-disjunction is literally the unequal distribution of chromosomes during cell division. You want things to go through disjunction, and non-disjunction is like not happening. You want things to go through disjunction. You want these homologous chromosomes to separate from each other normally. But here, what we see is that this homologous pair did not separate normally. So this is your non-disjunction right here. And then what happens is that you go through this, you keep going through this process, and this one has 1 too few chromosomes, and this has 1 too many chromosomes. And right here is an egg or a sperm that has 1 too many chromosomes. If you imagine it being fertilized with a normal sperm or a normal egg, you could potentially have 3 copies of chromosome number 21. This kind of trisomy, and trisomy literally means 3 chromosomes. This kind of trisomy, you only see it in chromosomes that have the fewest number of genes. So you can see it in chromosomes 13, 15, 18, 21, and 22. If I go back up to my chromosome numbers, 13, 18, 21, 22, and of course number 15. So reproductive age does tend to increase this, but it's not the only way that a child with Down syndrome or any other kind of trisomy might happen. We'll go into more details in this in lecture. So does that only happen to those autosomes, or can it also happen to the sex chromosomes? And it can happen to the sex chromosomes. I just used the word autosome, I realized, and I haven't defined that for you yet. So let me define autosome for you really quickly. Autosomes are the chromosomes with numbers, and sex chromosomes are the ones that are named with letters. So everybody has 2 autosomes that are exactly the same. Females have 2 sex chromosomes that are exactly the same, but males have 2 different sex chromosomes, in the case here, an X and a Y. sex chromosomes, and the rest of those are called autosomes. So it is possible, as I said, to have problems with too many or too few sex chromosomes. It's fairly common, actually, and this is an example of where you have one too few sex chromosomes. It's called Turner syndrome, and this is characterized by short height, a web between the neck and shoulders, a web of skin, underdeveloped ovaries, which can lead to sterility and some learning difficulties. But for the most part, somebody who has Turner syndrome probably might not even know it, except for they're on the short side. Don't go around looking at short women thinking they have Turner syndrome. They're not like super short, they're just in the normal range, but shorter than they might otherwise have been if they had had their Y chromosome or their other X chromosome. The next one is Klinefelter syndrome. And with Klinefelter syndrome, this could be, as with Turner syndrome, it could have been a non-disjunction with either the egg or the sperm. Same thing here. Either the egg or the sperm could have one too many sex chromosomes. It's distinguished by having 2 X chromosomes and 1 Y chromosome. And Klinefelter's people are male, but their testes are not developed properly. They have lower testosterone and are usually infertile. And there might be some feminine features associated with them, like a little bit of hips or maybe some breast tissue that's more developed. They also have longer limbs and are slightly taller than average. So Klinefelter syndrome. Please don't go around trying to detect Klinefelter syndrome either. The last two that I'd have to talk about are, there's not actually much big deal about these two non-disjunctions. The first one is called the XYY male, and literally having 2 Y chromosomes and an X chromosome. A little bit taller than average, a little bit more severe acne, and maybe something going on with the intelligence level, but it's really hard to say, and sometimes they're called super males. I hate that term. So we're just going to call them XYY males. The final one is XXX females, and they also may be sterile, but there's no obvious mental or physical problems. Sometimes called meta females. Hate that term. We're just going to call them XXX females. So the graphic in your book's a little wrong on the XXX females. This could be a problem with either the eggs or the sperm. But with the XYY males, it can only be a problem with the sperm because only the males have Y chromosomes. So that's what I've got for you for Chapter 8. Hopefully you enjoyed this lecture and learned something from it, and I will see you in class.